Welcome everyone. My name is Nicole Scott. I am the project manager for the Agricultural Genome to Phenome Initiative. AG2PI is funded by USDA NIFA, and this is our 26th workshop. Any of our previous workshops and our virtual field days, our conferences are all available on the AG2PI website at ag2pi.org. So feel free to go and check those out. Today, we'll be going through combined GWAS and TWAS using SVEN, and we have a couple of presenters today. Dr. Shomak Datta is an associate professor of statistics at Iowa State University. He received his PhD in statistics from the University of Chicago. His research interests include developing theory, methods, and matrix-free computations for high-dimensional problems. And we also have Dr. Vivek Roy who is a faculty member in the Department of Statistics at Iowa State University. Currently is serving 3 statistics journals: Electronic Journal of Statistics, Journal of Computational and Graphical Statistics, and Sakaya Series B. I'm so sorry if I butchered that, as an associate editor. His research interests are Markov chain, Monte Carlo, importance sampling, high dimensional data analysis, and model selection. So with that, I am going to hand it over to Shomak.
Hi, good morning, good afternoon, good evening to everyone, depending on which time zone you are at. I'm Shomak Datta. Thanks, Nicole, for introducing me. And I'm with Dr. Vivek Roy and our RA research assistants An Nguyen and Deboshi Chakraborty will be helping us with the session. So let me start. All right, so I hope you can see the screen, the presentation that says combined GWAS and TWAS using SVEN. So the organization of today's workshop would be as follows. We will first describe the methodology behind SVEN, followed by a demonstration of SVEN. And after around 1 hour, we will take a break, and then we will we will flip the workshop, and we will let you run code on the data set that we use to demonstrate. Now, the code and the data are available on this website. So slides are already posted, but they will be updated if there is any change. The data and code are posted here. So let me share this URL with everyone. As soon as I find the chat button. And yeah, I cannot find the chat button anywhere. So
Shomak, I will share it, don't worry.
Okay, thank you very much. Yes. All right. So yeah, and if you have any trouble downloading the data set, we can also share over Zoom. So we'll wait until the hands-on session to do that. All right. So GWAS and TWAS are 2 very important methods for studying, discovering gene associations with phenotypic variations or step association with phenotypic variations. So let me ask a question to Vivek. Or do you see this something here annoying, or is it just a slide that you see? Because I see like people are entering, or in the waiting room, I see a notification for that, a floating notification, and I'm wondering whether that's blocking out the slide.
That is not blocking the slides, but okay. I see those on top of the slides, but that's not, that's not blocking the slides. So I think it's okay.
Okay, sounds good. Okay, thank you. So yeah, we generally in modern problems, we have phenotypic traits, and we also have SNP data associated with those lines or varieties, and we have RNA transcript or gene expression data. Now we can do a GWAS, which is basically a regression of the phenotypic traits on the SNPs, or we can do a TWAS, transcriptome-wide association studies, where we regress the phenotypic traits on the RNA transcripts. And it is also it's a regression of the RNA transcript of the gene expressions on SNP provide us with the QTL mapping. Now, QTL mapping is not part of the workshop today. We will focus mostly on the GWAS and TWAS analysis. In practice, GWAS and TWAS are done separately, where the phenotypic traits are either regressed on SNPs or on the RNA transcripts. So they can be combined. These two separate analyses can be combined using the Fisher's combined test, but the statistical power to detect causal genes could be increased by combining the GWAS and TWAS through a single regression model. And that's what we're going to discuss next. So our example data set would be on a SAM panel. So it's a shoot apical meristem panel. It comprises of a small pool of stem cells that generate all above ground organs. That's the phenotype. In this SAM panel, we have 360 genotypes. We have close to 1.2 million SNPs and around 39,000 gene expressions are in transcript data. And for the SAM data, the goal is to identify the candidate genes associated with the SAM morphometric variation. This data set is published by Leibov et al. In the Nature Communication Journal. And this is kind of the visualization of the SAM morphology. So the SAM morphology, it could be small SAM volume, it could be intermediate, or it could be large. And we want to identify the genetic basis of what causes these SAM volumes to differ from one genotype to another. The statistical model behind combining the genome-wide association study and the transcriptome-wide association study is a high-dimensional linear regression model. So typically, we observe the trait values through field trials, and we compute the blues, the best linear unbiased estimators, or the best linear unbiased predictions of the genotypes. So for example, it could be the logarithm of the SAM values. So we have one observation for each genotype, and the vector of those observations, we call that Y. X is the covariate matrix of SNPs. So we have close to 1.3 million SNP markers associated with each genotype. So P1 here, N here is 360, and P1 here is close to 1.3 million. We also have a matrix Z, which contains the gene expressions. So here P2 is around 39,000. And the multiple linear regression model that we use to identify the SNPs that are important, or the genes that are important, is given by Y equals to mu naught times a vector of 1. So that is, the mu naught is the intercept term, overall intercept term. And then combine the effects of the markers, SNP markers, which is given by X times beta, where beta are the coefficients of the SNP markers, plus Z times alpha. So Z here is the covariate matrix of the gene expressions. So alpha are the coefficients for the gene expressions, genes. Plus we have random IID random errors. We assume that those IID random errors are generated from a normal or a Gaussian or normal distribution with mean 0 and variance sigma squared. So here, as I said, beta and alpha are the vectors of regression coefficient, and sigma square is called the error variance. The goal is to identify the best subset of predictors, that is the subset of SNPs and genes from all possible subsets of these SNPs and genes. If you're wondering how many possible subsets are there, the answer is 2 to the power P1 plus P2. So it's 2 to the power P1 times 2 to the power P2. And that's a gigantic number. So we need to identify from this haystack a few needles, a few SNPs, and a few genes that are supposed to be actually related to this phenotype or trait of interest. Multi-locus methods can consider multiple genetic variants. So it can consider the SNPs, all the SNPs together, all the genes together, and so account for the population, account for not only the effects of each other, but also account for the population structure and relatedness between all these individuals. And so it should have better potential to identify important genetic variants. In fact, in the statistical model that we have developed, we could prove theoretically that under certain conditions that either when your number of lines go to infinity or when your error variance sigma square it goes down, you are able to identify the truly important SNPs and the truly important genes with probability tending to 1. So let us first try to understand the model. So, what we do is a very hierarchical model. This is the kind of the workhorse behind SVEN. So here we have, we assume that, there are these latent indicator vectors, gamma and delta. So gamma is like 1,000,000 by 1 vector. And it indicates whether a particular SNP is important or not. So gamma j, we will say it's 1 if and only if the jth SNP is important. It's 0 if and only if the jth SNP is not important. Similarly, delta would be like a type 39,000 by 1 vector of latent binary variables, indicating the importance of genes. That is, delta j would be 1 if the jth gene is important. It would be 0 if the jth gene is not important. So we want to learn about this gamma and delta, which genes and which SNPs are important from the data. The generative model is given by this first line. So if you give me a combination of gamma, that is, if you say which SNPs could be important and which genes could be important, we would say that the response y is a Gaussian very random variable, normal random variable, whose mean depends only on those markers, SNP markers, and only on those genes that gamma and delta say should be important. These beta j's, the coefficients, they have what's called a spike and slap prior. So you can see that beta j given gamma, they're independent, normally distributed with a mean 0 and a certain variance. Now, what if gamma j is 0? If gamma j is 0, the variance is 0. So if the model says that the jth SNP is not important, then beta j would be a normal variable with mean 0 as well as variance 0. And in statistics, when the mean and variance are both 0, then the variable is actually just 0. It can only take the value 0. So if gamma j is 0, then the coefficient is shrunk to 0, exact 0. Okay, so if gamma j says the SNP is not important, then the coefficient is fixed at 0. So there's no effect of that particular SNP. On the other hand, if gamma j is not 0, that is, if gamma j is 1, then the beta j has a non-degenerate distribution. So, it says that, okay, the value of beta j is not 0. It can be any non-zero number with certain probabilities distribution. Similarly, for alpha j, the coefficients of the gene expressions. In this hierarchical setup, we assume that the intercept and the error variance, they have what is called non-informative prior. So the prior on the mu naught intercept is flat on the real line, and the prior on sigma square is proportional to prior density is proportional to 1 over sigma square. A priori now, we are for gamma and delta, a priori we assume that these are IID Bernoulli random variables. So that is, before seeing the data, we think that there is, based on our scientific understanding and experience, we think, okay, maybe W1 proportion, let's say 0.1% of the SNPs among all these things should be important. Okay. And so we say that, okay, a priori, these latent indicator vectors gamma, they can take the value 1 or 0, of course, but they can take the value 1 with a probability of W1. Okay. So the expected number of SNPs before we see the data, this is based on our scientific understanding, expected number of SNPs, we say, is the total number of SNPs times this number W1. And similarly, the expected number of genes that are important is the total number of genes times this fraction W2. And in this notation, if you want to, understand it further, mod gamma, this mod gamma and mod delta are the size of the model gamma and the size of the model delta. So it's not those are the number of ones in the vector gamma and number of ones in the vector delta. So number of SNPs that are deemed to be important, or the number of genes that are deemed to be important. Here, these numbers, the lambda1, lambda2, w1, w2, these are assumed to be known non-random functions of the sample size of the number of varieties, number of SNPs, and the number of genes. And these are typically elicited based on our prior understanding of the problem. And we will learn in different cases how to choose the values of lambda1 and lambda2 and w1 and w2. Right. Going forward, so let us try to understand this Bayesian hierarchical model. So we'll keep it small for pedagogical purposes. So for now, suppose that P1 and P2 are 2. So we have, let's say we have only 2 SNPs and 2 genes. So there are altogether 4 variables, 2 SNPs and 2 genes. Given that there are 2 to the power 4 many possible models, possible combinations. The first one is where gamma is 0 and 0, that is, and delta is also 0 and 0. So that is, this particular model says that it thinks that neither of the SNPs are important and neither of the genes are important, which corresponds to a model on the trait values as y, which is just intercept plus the error. As you can see, this model says that there is no effect of the SNPs or the genes. And we will learn later from the data whether this model is actually a good model, okay? The data will tell whether this model has high probability given the data. Similarly, let's look at another one. Let's look at this one. Gamma equals 1, 1, and delta equals 2, 0, 0. So this particular model says that, or thinks that, both the SNPs are important, but neither of the genes are important. So this corresponds to the linear model that the trait values is the intercept plus the effect of the first SNP plus the effect of the second SNP, but no effect of the genes plus the random error. And finally, the 16th model is where it thinks that both the SNPs and both the genes are important. So it corresponds to gamma being 1 and 1 for both the SNPs and delta being 1 and 1 for both the genes, and corresponds to a linear model where y, the trait values, are equal to the intercept plus effects of the 2 genes, 2 SNPs, plus the effects of the 2 genes, and plus the random error. Now we will learn among the 16 models, possible models, which one are more plausible once we see the data. And that is summarized through the posterior distribution. So we can find the posterior distribution given the data of these latent indicator variables gamma and delta. This posterior distribution is supported on these 16 combinations. On the 16 combinations of zeros and ones. And this is when P1 and P2 are small, which is true in this case, we can find this posterior distribution by analytically integrating out with respect to all these other nuisance parameters, the intercept, the SNP coefficients, the gene coefficients, and their variance. And in this particular model, we see that there are some models which are larger in the sense that they include more effects than the others. Now, if you have taken a course on linear regression, you would know that larger models tend to fit better. Larger models always give bigger R-squared values. Okay, so they tend to overfit the data. So in this case, the prior probabilities on W will shrink that. So we'll penalize them. So W1 and W2 are the prior inclusion probabilities of the markers or the genes. Now, if the model tend to include too many SNPs that are not important, then they will be penalized by this prior inclusion probability. So this W1 and W2, so these should be small. They will not allow an unimportant variable to enter the model. Okay. And they would not allow an unimportant SNP or an unimportant gene to enter the model. The theory that we worked out suggests that if you choose under certain assumptions, if you choose the lambda 1 and lambda 2, these are called shrinkage parameters. If you choose them as the sample size divided by the number of SNPs squared or the number of genes squared, and if you choose the prior inclusion probabilities as the square root of sample size divided by the number of SNPs or the number of genes, then they guarantee model selection consistency. That is, they say that you would as either your error variance goes to 0 or your number of lines goes to infinity, you would pick only the truly important SNPs and truly important genes. That is, both your false probability rate and false positive rates and false negative rates, both of them will converge to 0. So let's look at what we learned is that we can look at the posterior distribution of the latent indicator vectors gamma and delta, but that is supported on the entire space of, in this particular case, on these 2 to the power 4, 16 possible combinations. In practice, you will not have 2 SNPs or 2 genes. You will have hundreds of thousands of SNPs and thousands of genes. So this, we need some form of a summarization of these of these distributions. They can be done in many ways. We consider here 3 methods. The map model. The map model is the model which is a combinations of SNP and genes that is most probable given the data. Okay, the combination of SNPs and genes, the model with the highest posterior probability. That's the most probable combinations given the data. There is also the weighted average model. So weighted average model, it says you look at the posterior probabilities of each of these models, possible combinations, and you just take the sum. So you find the posterior probabilities, and you weigh each model by its posterior probability and add them up. That's a weighted average model. And the last one, which is the MIP, I think is probably the most important. this gives the marginal inclusion probabilities of each marker and gene. I should have also included the genes. So the marginal inclusion probability, it's the probability for any SNP or any gene is the probability that that SNP is important or that gene is important given after accounting for the effects of all other SNPs and all other genes. This should be used for getting what are called the hits on the markers or the genes. Let's look at a particular example, a small toy example. So let's say we have the synthetic data set generated from a model, from the model with n equals to 60 lines. So in a typical plant science problem, the line number of lines, the population under consideration would be kind of fixed. So let's suppose there are 60 lines for now, and that there are two SNPs, and we are interested, looking at these two SNPs and these two genes because, we can then summarize the posterior distribution. And let's say only the first SNP is important and only the second gene is important. The first SNP has a coefficient of 0.1. The second gene has a coefficient of 0.4. And the other SNP and gene, they have a coefficient of 0. So they're not important. So this is how the data was synthesized. And we will consider different values of the error variance. Now, this error variance, the sigma square, is connected in a plant science study, is connected with how many reps you do on the lines. So if you do more reps, then your sigma square would be smaller. That's the stat 101. So the standard error of the genotypic blues that is related to sigma square, it decreases with increasing number of reps. All right. So here, and x1, x2, z1, z2, these are generated, synthesized from a normal distribution just for pedagogical purposes. All right. So what we see here are the posterior probabilities for three different cases. In the first case, sigma is a bit large. So the error variance is 1.4. In the second case, the error variance is 0.5. So it's like, you have more reps. And in the final case, your error variance is, error standard deviation, sorry, is 0.1. So if even more reps on the varieties. All right, and here the first, second to fifth column gives you the combinations of gammas and deltas. As we discussed, there are two SNPs and two genes, so there are 2 to the power 4, total 16 possible combinations. The first one is that what sometimes people call null model. It's a model that thinks none of the SNPs and none of the genes are important. So dot here means 0, and 1 here means, well, that's 1. So that particular SNP or gene is important. Now, when you see that when sigma is very large, it's 1.4, the posterior distribution is not concentrated on the true model. Now, the true model is its 10th model. Why? Because it says that the first SNP is important, and the first gene, so the second gene is important. But the second SNP and the first gene are not important. So this is the true model. This is how we generated our trait values, synthesized our trait values. Oh, sorry. Now, we see that that has a very small problem. So here in these few columns, the dot means they are very small numbers, less than 0.001. Okay. We see that most of the posterior probability is now concentrated on the null model, where none of the SNPs and none of the genes are important. So given the data generated from a very large sigma square, large error variance, we don't get much hits. We see that there is this one model, model number 9, which thinks the second gene is important. That does get some descent posterior probability, which is 0.38. But most of the mass is concentrated at the null, on the null model, and a little bit on this particular model number 3, which is also a wrong model. It says it thinks second SNP is important. Only 1% of the mass is there. When you increase the number of reps, let's say your sigma decreases to 0.5. Now we see that one of the SNPs one of the important genes, that is, Z2, that gets decent posterior probability. That gets almost all the posterior probability. So 97% of the mass concentrates on the model that says none of the SNPs are important. The first gene is unimportant, but the second gene is important. Okay. And this is because, the first SNP has a relatively smaller effect size. The coefficient is 0.1 compared to the coefficient of the second gene, which is 0.4. So this effect of the SNP is small. Although it's non-zero, it's small. So which is why it's not detected even when sigma is 0.5. But when you increase your number of reps further, that is, if your sigma reduces to 0.1, now we see that the 10th model, which is the true model, gets the highest posterior probability. So almost all the posterior mass concentrates of this true model, which correctly says that the first SNP and the last gene are important. Okay, so the takeaway message from here is that you won't be able to detect SNPs or genes with small effect size unless you have a lot of reps on the genes. Okay. Now, here's another example. It's when, what I often see in the GWAS data sets or SNPs, TWAS data sets, that there are often duplicated SNPs that are highly correlated or are duplicated, or genes that are highly correlated among themselves or are duplicated. And what's the effect of those? So the thing is that duplicated SNPs or gene expressions can hurt discoveries. Here's how. So here we say we fix, I think sigma is 0.5. Now, the way we generate the data, it's kind of the same model from before. So we have the first SNP that is important. We have a gene that is important. But now we set Z1, the expression of the first gene, to be exactly the same as the expression of the second gene. Okay, so Z1 and Z2 are identical. And here's the effect of the shrinkage. Okay, so if we see that, if we use small shrinkage, so let's say lambda is 0.01, it puts the posterior mass divided between most of the posterior mass divided between two models. Which are the models? This particular model says, well, the first SNP is important. Let's keep that separate. Both of them has the first SNP important, right? But the other case is either gene number Z2, gene number 2 is important, or gene number 1 is important, okay? Because Z1 and Z2 are identical, this one, when lambda is small, it puts equal mass. It cannot decide between which model to pick, okay? It puts almost equal mass on those two models. If we increase lambda to, let's say, 0.1, we see that some of the mass on those two models reduces, but the mass on this model number 14, which says the first SNP is important and both the genes could be important, the mass on that increases. Now, because Z1 and Z2 are identical, I would think that, this is the correct model because Z1 and the two genes have the same expression. So biologically, in this data set, both are supposed to be important. And so the posterior concentration on that model increases. Finally, when we increase lambda to 1, use higher shrinkage, we see that the most of the posterior mass now concentrates on this particular model number 14, which thinks that the first SNP is important and both the genes are important. Okay. So in general, if you use small lambda, then the total probability gets divided into equivalent models. Okay. And same thing happens when Z1, Z2 are very highly correlated. In this particular example, Z1 and Z2 are exactly the same. The two gene expressions are exactly the same. But in practice, they could be highly correlated and the same story would hold. In this kind of cases, what we learn is that we should use a high shrinkage in order to identify all the genes, both the genes, okay? So comments or some takeaway messages is how to, it's on, the first one is on how to choose these shrinkage values, lambda1 and lambda2. Sorry, I'm seeing the bit messages, but okay. So it is, if your trait is complex, that is, if you think that your trait is controlled by a lot of SNPs or a lot of genes with individual small effects, right? Then it is as advised to keep the lambda1 and lambda2 high. Somewhere in the previous example, we saw that 1 helps. Somewhere in the values 0.1 to 1 would help. That was also our understanding in the paper. But it is also a good idea to thin the SNPs, because there are so many SNPs. It's advisable to thin the SNPs to reduce the effect of the slow LD decay, to make sure that that they're not highly correlated SNPs that steal the effects of one another. Okay. And of course, we do quality control on the SNPs and the genes data that are always a part of the GWAS or TWAS analysis. So those are crucial, but those are not within the scope of this workshop. So generally 5% of the SNPs are only SNPs with 5% or more, or certain minor allele frequency is capped at threshold. Some form of minor allele frequency thresholds are used to quality control the SNPs, or outliers of genes are tackled. So those quality controls are needed. And a final note here is that you can also use SVEN. SVEN here, we are talking about combined GWAS and TWAS analysis, but you can also use SVEN to run only GWAS or only TWAS. So if you only pass the SNP matrix, it will do GWAS. If you only pass the gene expression matrix, it will do TWAS. And if you use the gene expression as one of the gene expression as the response, and the SNP matrix as your SNP matrix for prediction, or sorry, as a covariate matrix, then it will do the QTL mapping. So those are also within the scope of SVEN. And these are the two papers. The methodology is published in this paper by Lee et al. And the SAM panel comes from the paper by Leiboff et al. All right, so before we go into demonstration, I would like to know if there is any questions. I cannot see the chat. If you're welcome to ask the questions on chat, or you can also, you're welcome to wait till the the hands-on session or to ask the questions. I don't see the chat, so if
so Sumuk, I see the chat, and I am replying to the messages there.
Okay, thank you.
Yeah.
Can you ask the question?
Yes, go ahead. Let's read one.
Yeah, thank you. I'm just wondering if we can use it in situations where we have a few of the data sets lacking gene expression data. That is, when we have the phenotype, we have the SNP data, and some of the samples are lacking in gene expression data. Can we include those samples in the analysis?
Not yet. That's a very good question. So the question is whether, you have, let's say we have this 316, actually in this data set also we had 369 lines. We actually had 369 genotypes, but we didn't have gene expression data on 9 of those genotypes. So we had to drop them. So not right now, but we are working on extending these methodologies to the case where you may have missing gene expressions. So yeah, that's probably going to be the discussed in the next year's workshop.
Yeah. Thank you. In this case, for maybe complex traits, do you have an idea of a reasonable sample size that you can use, since we need to have both phenotype, genotype, and the expression data?
Sample size, or would you mean number of varieties or number of
sample size.
reps?
No,
Sample size. So generally, that's a very good question and very tough question. I don't know the answer to that. You definitely need at least 2 reps to be able to kind of, get the blues. And so it also depends on other sources of nuisance variabilities. So oftentimes these agricultural, the trials are done on fields. The fields have spatial variability. Those are accounted for. So it depends a lot on the other sources of variations. So yeah, I don't know how many. I cannot give you an answer to that. But what we can, but the theory says that if your sample size is small, then more sample is always better. Let's say that's one way to put it. More samples are always better, and the more samples you have, the more confident you can be on the discovered SNPs and gene expressions, on the hits, basically.
Thank you.
You're welcome. All right, so let's go forward. So it's a little demonstration. So we use the SAM panel, but the SAM panel has close to 1.3 million SNPs and 39,000 genes. So we are not going to use that entire data to demonstrate because that analysis takes a lot of time. We're going to do a bit of thinning. So we are going to select 10,000. We have selected 10,000 SNPs from all these 1.3 million. We did some quality control. SNPs with minor allele frequency less than 5% are dropped. Major alleles are coded as 0. Minor alleles are coded as 1. Missing values were replaced by the probability of being a minor allele. And we store this matrix in a sparse matrix format. So we will see in a bit on R. And we selected 2,000 genes out of these 39,000. And for each gene, we pre-process them. So the gene expressions less than 5 percentile for each gene, the values that are less than the 5 percentile for that gene are set to 0. The gene expressions bigger than the 95 percentile are set to 2. And the remaining values between 5 percentile and 95 percentile, they are linearly interpolated to the interval 0 to 2. Okay, so all these gene expressions are scaled to 0 to 2. This is what the practice this is also the practice in Leibov et al. But this is not the hard and fast rule. There are other ways also to scale gene expressions or pre-process gene expressions data. And the phenotype values Y are generated synthetically. So the data and code are available at this particular website. Vivek, if you could share this URL on chat. So, and now I will switch to the RStudio, to R. And okay, I hope that you can see my.
Yes, it looks good.
Okay, thank you. All right, so we use the function SVEN is included in an R package Bravo. So all right, so this is available on CRAN, the Central R Archive Network. The current version is 3.2.1, and the binaries are available for Windows and Mac, so it should be easy to install. All right, so okay, so if you don't have Bravo installed, or if you have earlier versions of Bravo installed, you should, so this version was released like just last week. So you should install that packages and Bravo to install this package on your computer. So we will use the matrix package to do the sparse matrix algebra, and we will use Bravo to run the GWAS and combined GWAS and TWAS. If you inflate the files, which it will do in the hands-on session, the matrix is the SNP matrix, gene expression matrix, Z, and the phenotypes are given there. So the SNP matrix is a 360 by 10,000 matrix. The gene expression matrix is a 360 by 2000 matrix, and Y is a 360 vector of length 360. So Z is stored as a native R matrix. It's just a matrix, it's a dense matrix, but X is stored as a what is called a sparse matrix. So the fancy name is DGC matrix. So it's a double precision column major format matrix now. So it only stores, let's look at the first 10 by 10 submatrix of X. So what it does, it stores only the non-zero values. So the minor allele values, which are 1, and the probability, for some SNP, it's a probability that's the minor allele. Only those are stored, but the major alleles, which are coded as 0, those values are not stored. So that reduces not only the storage requirement in RAM, but also makes the computations faster, because if the program knows that those values are 0, we need multiplication, so it can do the arithmetic. It doesn't have to, 0 times any number is 0, so it doesn't have to do those computations explicitly. So the 0 values are all dealt with without any computational cost. All right, so we do the combined GWAS and TWAS analysis. So let's run it. It takes a bit more than 1 minute to run. So while it's running, we will explain. So here we pass the SNP matrix and the gene expression matrix as a list. Okay, so list of 2 matrices, X and Z. If you pass only X, it will do the GWAS only. If you pass only Z, it will only do the TWAS. But because I passed both the X and Z as a list, it will do the combined GWAS and TWAS analysis. The second argument is the vector of response Y, the phenotypic traits. Now this is rest of the arguments, so as follows. So lam here is the vector of lambda 1 and lambda 2, and I think the SNPs could be the trait could be complex. So I'm using shrinkage values of 0.1 and 0.1. Okay. If you don't think your traits are complex, then you can use the default values of lambda, which are n over p squared. So those are smaller. There's this argument, 10 is the default value, so that's fine. Here I use temperature. So the SVEN is an optimization algorithm, and it uses a temperature schedule to search among all the models. And we use 10 temperature values because here, so the temperatures are used because sometimes this high probability models can be separated by values in probability. And in order to facilitate SVEN to jump from one high probability model to another one, we do use this kind of tempering. And the last argument is 10. True. So this is verbose. Verbose equals to 2 just tells you which temperature SVEN is more working at. So there are 10 temperature values, so it should give 10. It just helps me see that something is happening. It's not just it's just stuck at something. So just something to keep me interested. All right. So once we fit the model, we can use this function MIP.SVEN. So MIP stands for marginal inclusion probabilities. So we can use the MIP.SVEN to get the hits. So let's compute that. Now, typically there would be a lot of hits with small probabilities, right? So let's see how many. So hits is a list of. In RSDF format, it's a list of 2 entries. So the first one is the hits on the SNPs, and the second entry would be the hits on the genes. Now this says that there are close to 78 hits on the SNPs. Okay, that's quite a few. Let's see that. I didn't want to print it out in the entire document. So you see that these are the hits. And the marginal inclusion probabilities, most of them are pretty small, some are pretty big. So this one is like 100%. So it's very sure that that probability, that particular SNP is important. SVEN thinks that this particular SNP is, has a 100% chance of being important, numerically speaking. So it's very close to 1, actually. You can sort them to see how many. So it seems like the 6 markers have very high inclusion probability, marginal inclusion probabilities, and the other hits, they are pretty small, all less than or equal to 6%, right? So I didn't want to see all of them, so let's we can rerun SVEN to say, okay, I don't want to see all of them, just show me all those hits with a marginal probability, inclusion probability of more than 20%. Okay, so let's do that. And now it's, I think, a more manageable number, as we saw that only 6 SNPs had a marginal inclusion probability of close to 100%. How about the genes? So let's look at genes with hit probability or marginal inclusion probability of 20% or more. And we see that there are 5 genes that have marginal inclusion probability of 20% or more. You can change the threshold according to how you want. I mean, you can keep it as is, because then you can, if you're looking at the results on an Excel file, it should be easy to see. All right, so here we see that these 3 genes, they have, SVEN is very sure that those are important. But for the 2 other genes, SVEN thinks that, well, there's close to 5%, 50% chance of them being important, right? Now, why is that so? It's because, so we can look at these particular genes and we can look at the correlation between those 2 genes. And we see that these 2 genes have more than 99% correlation between the 2. And as we saw that, when that happens, then basically the posterior probability gets divided between those 2 genes, okay? So that's about it. So it thinks that, all these 5 genes are good. SVEN thinks all these 5 genes are good hits. Now, if you also look at the gene numbers, they are, you see the last 3 digits, the first others are all the same. So these 2 genes are very close to each other. So this one has 790 and this is like just 10 units apart. So it's 800. So they're very close to each other in the chromosome, which is probably why they have a very high correlation. So biologically, both should be important. So that's about it for this demonstration. And then after this, you can go to the database, to a genetic database to identify identify the genes and SNPs. So yeah, that's about it. All right. So yeah, if there are other questions.
Yeah, I have one question, if I may.
Yes, go ahead.
I was wondering how you take relatedness and population structure into account in your model.
It's a very good question. So how do we take the relatedness and population structure in the model? So the population structure and relatedness are often used in other methods, such as which are single loci or even multi-loci, but they do not scan the entire genome at the same time. So let's see. Right. So oftentimes in many methods look at one or a few SNPs at a time based on KB on a window. Okay. And because of that, so it doesn't really account for the rest of the SNPs unless the principal components are used to account for the rest of the SNPs. So in other models, the principal components are typically used to account for kind of, the remaining SNPs and the population structure and relatedness. But here, because we use the multiple linear regression model on the entire SNP and entire gene at the same time. We do not have to explicitly account for those population structure or relatedness. Those are automatically accounted for in this model. Now, it is possible to extend this model further to, for example, if you do thinning of the SNPs, it is possible to include the principal components to extend this model, but they are not yet within the scope of this function SVEN. But that's also something we are working on. In case you are interested to use SVEN, but not with the entire SNP, but only a subset of the SNPs. Does that answer your question?
Yes, I think so. But I guess if your trait is related to population structure, you will have many SNPs related to the traits, and so the signal will be spread across all these SNPs, and so it will be taken into account in this way, right?
Right. So yes, it would be taken into account. If there are many SNPs now, kind of the false discoveries would be penalized by these prior inclusion probability. So if there are a lot of SNPs that kind of define the population structure, they will be penalized.
Yes. Okay, I see. Thank you.
You're welcome.
And I have one question.
Yes, go ahead, please.
With this type of analysis, what kind of genes you think you're targeting? Genes with minor effects or genes with major effects?
That's a very good question. So surely the genes with major effect will be included, but question is whether the minor genes with minor effects, will they be included, right? So it depends on how many reps you had in your data. So as we can see here, a SNP with a small effect compared to the gene, that was not the same conclusion would hold with genes with small effects or other SNPs with small effects. With small effects, they may not be discovered unless you have lots of, sufficient number of reps to include them, to discover them. Does that answer your question?
Yeah, that's fine.
Yeah, it's now.
It's like, to me, it's like association analysis type,
Yes.
no?
It is indeed, yes. Vivek, did I answer that? Is there anything you want to add?
No, that's fine. That's fine.
No, I was asking Vivek, the other presenter. So my colleague,
yeah,
yes.
I think yes. Yeah. So you can, as you have mentioned repeatedly, by using these hyperparameters lambda and w, you can try to recover those those genes with
Yes.
small effect size. Yes. But yeah.
Yeah, we can use the
That's
hyperparameters.
also the science has to come in there, whether few genes with higher effect size or also like what kind of traits we are dealing with, as you mentioned, whether it's a complex trait, and yeah.
All right, thank you.
You're welcome.
So, Dr. Dotto, I have some questions.
Yes, go ahead, please.
So, can I proceed with the question?
Yeah, yes, please. Sorry, can you hear me?
Yes,
Okay,
yeah, I
yes.
can hear you now. Yeah, thank you for the outstanding presentation. So, say when you do the SNP call, then you got a array of SNPs, and then the transcriptome analysis will give you the ideas about the gene. So my question is, how and when you are estimating the effect size of the SNPs and the effect size of the gene, and then incorporate in the model?
Yeah, that's a very good question. So, in the model, how are we incorporating the effect sizes, right? And that's
Yes.
through this, though effect sizes are incorporated in the model through this distribution on the coefficients. So each SNP has its own coefficients, let's say beta J's, and each gene has its own coefficients, alpha J's. And it is possible. So yes, the beta J's and alpha J's give the kind of effects. Now, if the gene is important, then the effect, the model says that the effect is Gaussian, normally distributed with a non-zero variance. It's shrunk a little bit, but not too much. But on the other hand, if the SNP is not important, let's say gamma G is 0, then the effect of that particular SNP is shrunk to exactly 0. So that's how the effect sizes of the SNPs and the genes are incorporated in the model. Now, there are many other ways of incorporating the SNPs, the effect sizes. But yeah, we are also kind of working on, there are many models, right? All models are wrong, but some are useful. So we are working on also incorporating other types of effect sizes in the model.
And in your combined analysis, when you are combining GWAS and TWAS, how does that temperature add values to it? Like I missed that part.
Oh yeah, it has to do with the kind of stochastic shotgun search algorithm that we employ. So there are lots of models, right? There are actually 2 to the power P1 times 2 to the power P2 many models. Now, many of these models, as you can see, most of these models are not important. Like most of these models say that the genes are not many genes are actually important. Not many SNPs are important. But actual good models that includes the truly important genes and the truly important SNPs, they can be very far away from each other. For example, they can be like, I don't know how to say it, but it's a 2 to the power P model space, and they can be like different parts of that model space. Now, these stochastic search algorithms often have a drawback that they tend to get stuck in a local mode. They find one model that has a good probability and doesn't want to go away from it. Okay. But there could be other models. So what we do is use this temperature. So when you raise the temperature, you kind of flatten the posterior surface. When you flatten the posterior surface, then it becomes easier for the search algorithm to jump from one model to another high probability model, across the values. Okay. So yeah, that's we can talk more about it later.
Thank you. So thank you very much. Yeah, got some ideas.
One other thing I would like to mention here is that the combined GWAS and TWAS analysis. Now, there are many SNPs that work through what is called the mediation analysis, right? The SNPs can actually affect the genes, and the genes in turn actually change the phenotypic values. And the power in the combined analysis is that, so if many SNPs are affecting a gene, then the model would prefer to select the gene because it's one gene explaining the effect of many SNPs. And so that's how it would, improve the identification of the causal genes, actually.
Hi, Samak. Yeah, I have a question related to that. So in the combined TWAS, we should expect more either SNPs or genes associated with any trait if we run separate individual GWAS and TWAS. Am I right?
Okay, that's a very good question.
So yeah, because I'm thinking like, how is like biologically integrating all those SNPs and all those genes? So like, are we finding more SNPs because maybe one SNP is affecting in trans many genes, and then can the model be able to pick that information?
Yes, that can be. I mean, yes. So that would be the case. It would pick up the gene. Now, whether the question was whether we get more hits with this method, but that's not necessarily true. what this method guarantees is that not guarantee. I mean, in many of the guarantees are in the asymptotic sense. In a finite sample, we don't know how big the error variance is, right? So, in the like asymptotically, the guarantee is that the discoveries are going to be truly causal. So whether we get less, it is possible that we get less hits in this method because individually there could be more false positives. But the combined analysis reduces the false positive rate so that we get less hits. Or it could be, it could also give more hits if there are true negatives, true positives that were ignored, in the separate analysis because the effects were not completely or fully accounted for. So
okay, so yeah, the takeaway message is like the hits or the genes that we are getting, we are more confident that those are having an effect. Okay.
yes,
thank you.
yes. All right, these are great questions. So let's take a break. Let's take a break for a few minutes, and we will reconvene. And meanwhile, if you could please install get your RStudio up and running, and install the package and download the data, put them in the same folder. Install the package Bravo from CRAN website. That would be great. If you face any issue in downloading or installing or anything, okay, please let us know. We'll be very happy to help. Hello, everyone. Welcome back. It's 25 past 11 Central Time in the US. So we'll resume. So we have two research assistants with us, Deborshi Chakraborty and Nguyen. They will be helping you out with the hands-on session. So if you have any questions, please share on the chat, and then Deborshi would be happy to help you out. I'll be here too. If you have any question for example, if you want to, ask any question that uses your data and are sensitive of nature, we have breakout rooms where we could talk in private. Let us know, please.
So everyone, I am just going to reiterate what Dr. Datta said. So I'm going to show you just what the files are, and maybe I'll just briefly go through how to run it in R. And after that, if you have additional questions, you can always ask. So let me share my screen. So I can share my entire screen. Okay, can anyone respond if you can see my screen?
Yes, I could see it.
Okay, thank you. So if you can just Google Somak Datta, and you can go to his website, where you will have a link to his personal homepage on the same server of Iowa State. And in his personal homepage, you will have a tab on the top called Workshops. So if you click on that, you will just get the flyer of this workshop, the slides represented, and a link to Box. And if you download the whole folder, there's an option to download. I don't think you need to sign into Box. Oh yeah, that's perfect. You don't need to actually sign into Box to download the folder. So it will be downloaded in a zip format. So you can just extract the files from this zip thing, and you should get 4 files there. One is an HTML file in which the data description, briefly the method, and the R code is completely written along with the output, which is we try to make it as far as self-explanatory as possible so that you don't face any difficulty when you look it back. And you should have 3 more files where those 3 files are X, Y, and Z. So basically, if you look at the function SVEN, you have to, as a data, you have to give 3 inputs, that is the response vector Y and the matrix containing the gene expressions, that's C, and the matrix containing the SNPs, that is X. So you just download those 3 files from, like extract them from the zip folder and store it in the same location where you have your RStudio installed. And then we are going to run the codes that are demonstrated here. So it's pretty simple. You have to install the package Bravo and Matrix. Currently, this library method is not absolutely needed. So to install Bravo, either there are 3 ways of doing it. So in your RStudio, you can either go to this packages option and then install, and then type the name Bravo here. And this is a really manual way of doing it. You can click install and Bravo will get installed. Similarly, for Matrix, you can do the same thing. Otherwise, you can just do this require Bravo package or require Matrix package. And if you have them installed previously, you can just call in this method, library Matrix or library Bravo. So far, so good with me. If not, please let me know. Till now, I talked just about installation and setting it up in R, nothing else. Okay, if anyone doesn't raise any confusion or question, I will proceed. I think if we have so Nikki, we provided you the RDS file exactly. It will be better if you download that. Is there any difficulty you are facing? Oh, for future, I think there will be some straightforward method available in Google. And if R supports it, then you can just read it in a data frame and export it in whatever format you want. So that is just read dot something and write dot something, if R supports it. But for the time being, let's not talk about this because like converting files that is not the scope of this workshop. If anyone has any problem in like downloading these files and getting to RStudio and installing the libraries till now, you can let me know. Okay, if not, then so this is a very easy way to read RDS files in R. So you can just read like this. Let me call this X. You can just do read RDS X workshop, and let me call this Z. So the same way, and let me call this Y. Okay, it's done. And just to check how many observations we have 360 observations. So let me do XZ here. So in SVEN currently, if you want to just know that how the, not the underlying mechanism, but how the SVEN function works in R, it's very easy to see the help page of R. You can just do this question mark, SVEN, and you can press Control Enter. By the way, Control Enter is a shortcut how you run codes in R. You can do this thing too, like the run option here, or you can just do Control Enter. So here you can see that what inputs does it take, and oh, this is the oldest one, I think. Oh no, it's fine. So this X is a list of two matrices. So for our example, the first matrix is the one which contains the SNPs, and the second one is the one which contains the gene expressions. And Y is the response. These things are pretty simple. So W, lambda, Shomagda just described the theory behind it. So by default, it chooses the W and lambda like this. But if you want, you can play around with those values. Like in this code, we have chosen both the lambda 1 and lambda 2 to be 0.1. And ntemp, it takes 10 by default. But again, you can choose to play around with it if you think that you really want a bird's eye view of the posterior surface. And it will also say like a little bit of math which is involved in this and what output it gives. So let us keep things simple. So let me call our model fit. So what the SVEN function inside the Bravo package does, if you just specify the two matrices and the Y, it will give you a lot of outputs, but let us run and see what we are interested in. So you can see, if you can see my cursor here, it has started running. So since we have provided 10 temperatures, it will iterate through 10 temperatures, and then let's see what happens. So as demonstrated, our goal is to discover the important predictors, namely the important genes and the important SNPs, which explain the variability present in the response. Let's see what happens. And we have given you an HTML file, so you can just go to this HTML file, which will be open in some browser of your computer, and you can just copy and paste the codes if you want. Okay, so 1, 2, 3, 4, 5, 6, 7, 8. For 8 temperatures, it's running pretty fast. And it's almost done, I think. One more to go. Okay, there we are. So our model is fitted. Let us see what things we can extract. So if you type fit and $1 after that, we can extract any output the SVEN function returns. So we told that we have different kinds of methods to get the posterior probability. This map is the highest posterior probability model, and this WM is weighted average thing. So if we see this model, these are the just column indices. So basically our X that had 10,000 columns, and our Z that had 2,000 columns, the data part of the data we used for this workshop. So you can understand that these are the SNPs which are selected, that's the column numbers, and these are the genes that are selected. So similarly, we can do for model.wam. So it should be pretty similar. And now if we run this mip.sven, we can let us see what it returns. So it's so yeah, it returns the which of the SNPs was selected, the column names, because the people who are working in biology are really interested in the column names. So the names of the SNPs and their marginal inclusion probability. And for the gene expression, it does the same. It returns the name of the gene expressions and their marginal inclusion probabilities. So let us see the dimension. It's dollar. So for one, it's the SNPs. So it has returned 93. It's just too many. Similarly, for gene expressions, it has returned 51. That's also too many. So we can put a threshold, like we want only the top 20%. So if we run this, did I make some? Oh, sorry, there should be a comma. My bad. Yeah, that's good. Okay, now if we look at the output, it's just 6 SNPs and 5 gene expressions. So let us see. So for the gene expressions, yeah, if you check the dimension, it's just 5. And for the SNPs, it's just 6. Okay, so let us look at the gene expressions. Look at the SNPs first. So hits double bracket 1. That's how we extract elements from a list in R. So see, all the variables it has selected has really good marginal inclusion probabilities, that is of 1. So it's fine. And now let us see the hits and extract the second thing from that. So the 3, 4, and 5, like these 3 genes have really like asymptotic marginal inclusion probability of 1. So these are fine. But these 2, you can see that they have almost same, not 1, but almost same inclusion probabilities. So we suspect that they might be highly correlated. So we can just check the correlation between them. So from the Z matrix, we can just check the correlation. So my matrix is small z, I think. And if we check, we can see that the correlation between them is very, very close to 1. So that's why both of them are selected with equal, because the inclusion probabilities are distributed around them like that. So we can choose any one of that if we want, depending on domain knowledge experts. So yeah, this was a brief demonstration of the thing. And now let me look at the questions you have. Okay, someone is asking that error in cross prod, okay, yeah, because we have used cross prod inside. So to use cross prod, I think you need the matrix package where you have to be careful that the M is in caps. Okay. Error in base call means, same for the call means, I think you have to load the matrix package. Were you in the same directory where RD? Yeah, you have to be in the same directory where the RDS files are. Yes, then only the read RDS will work. Otherwise, you have to specify the whole path of the directory. Are you still facing problem in getting the running the call means or cross prod, those functions? If you have the same error, please let me know. Let me stop sharing and okay. So after installing the package matrix, you have to call that package in your R session, like library matrix. Like installing that is not enough. You have to call that I want this package running in my session. So have you done that? Have you called the library? If you have not, just call the library once and then try it.
Yes, I installed it, and I also call the library as matrix.
Hmm.
But still the same error.
You are getting the error in cross-prod?
Yeah.
Okay, that shouldn't be the case though. Okay, okay, I'll get back to your question. Okay, Ali, what do you mean? How do you format the input files? What formatting we're talking about?
The gene expression, values, for example, the TBM, once you have how do you format them to the RDS for, format that you guys, use for the 7.
So once we have a data frame, we can write that file or export that in any, like several formats we want: Excel, CSV, RDS, text. There's a command called write RDS or write.csv like that in R.
Okay. Thank you.
So people facing problem with cross-prod. Let me see. Cross-prod should be in base R only. Yes, it's in matrix definitely, and it's also in package base, and it's in the package matrix. Yeah, it's in the package matrix, so it should work. Yes, it can be because of the variable names. Like if you're putting the wrong variable names. Okay, you have checked. Both the variables names are in uppercase. Okay, so Shumon, it will be good if you can share your screen and show it to me. What exactly? I'll stop sharing my screen. And
can you hear me?
yeah, I can hear you.
Okay, thank you for giving me the opportunity. I'm sharing my screen
Yeah, sure. Oh, Nikki, we have I have already shown you that you can find the code.
So you can you can see my screen.
Yes, I can see you. I can see.
Okay, yeah. First, I have called the library matrix as
Yep.
well as the Bravo function,
Yes.
and then I have set the working directory where
Okay,
I have stored all the data you shared earlier, and then run the code one by one and check also the dimension or length of different variables as well as the classes. And finally, when I have run the hit, I got this error. I don't know why.
Oh, okay. Oh, okay. I think it's the older version of Bravo that is installed in your computer. Maybe that's the problem.
Okay, I just have installed the Bravo.
Then it shouldn't be, because the binaries are updated for every version of R. So okay, let me see. Error in base call means X must be an array of at least 2 dimensions.
Okay. Does it creates any problem if the matrix version is older?
I don't know, but you can try doing that once. Like you can just tick off the matrix from your bottom right panel and install that again.
Shumon.
Okay. Yeah, please.
Devorsi, sorry for interrupting.
Okay.
Can you just try, instead of putting the SVEN list XG, can you just pass X,
Oh,
simply
yeah,
X.
yeah, I can, yeah, sure. N and G, X and
No,
G?
just X.
Just
Just
X.
X.
Oh,
Yeah.
only the X.
Yeah, that's it. Can you
Like-
run that?
No, even it's not. Yeah, even it's shown error.
Okay, can you?
One minute. For the lamb, you have to specify just 0.1.
Oh, yeah, sure.
Yeah, because there's two.
Yo, I didn't put the matrix then, and yeah, it's
Yeah, now
working
it's running. Now it's running.
Yep, it's working.
1 minute.
Yeah, it works now.
Okay, so the just you can come to your R console and type $SVN.
SVN. Yep.
Question mark
Sorry, yeah,
Sven.
sorry. Single or double question?
Just a single one.
Okay.
Yeah. Now can you just scroll down a little bit more? Okay. So yeah, I think it's the older version of SVEN which doesn't support two matrices, because if you see the description of X, it shows the N cross P matrix without intercept. So this is the older version of SVEN, I think, because the newer version will say that it needs a list. So I recommend you to uninstall SVEN and install it again.
Okay, let me do it again and come back to you. Okay, thank you for your time.
Yep. So before running, you can just check the description like that from the help page, and it will tell you that it needs a list. That's how you can verify that the new version is installed.
Okay, thank you. Got it.
Yeah. And always let us know if you face further problems. Yeah, definitely you can share your screen. I can't really pronounce your name. I apologize for that. But yeah, definitely you can share your screen. Okay. What's your problem again?
Can you see my screen?
Yeah, I can see your screen. I can see the HTML file.
Yeah, it's I run it, the library, the Bravo, it works, and even I installed the matrix.
Okay.
And the same, it looks correct, and the X, Y, X.
Okay, I am interrupting a bit. I can see the HTML file, not your RStudio.
Okay. Sorry.
Oh, now I can see it. Yeah, thank you.
Okay, thank you. So Bravo, it works, and even the matrix also it works. Here are the XY files, and this one is the.
Yep.
Then shapes like this.
Crossword XY. Okay, again, what? It is an error in crossword. Yeah. Okay, can you just run that line 13? And can you show me? Show me the X description of the X matrix. Just although this shouldn't be a problem. Oh, it's fine. The SVEN is fine. It's the updated SVEN only.
Okay.
But I think there's some problem in matrix multiplication. Okay, can you go to the packages tab of your bottom right window? Yeah, it might be the version of matrix. It might be the version of matrix because SVEN uses the latest version of matrix.
Okay.
Can you go to the packages panel on the bottom right window?
Yes.
Yeah, packages. Yeah. So you can scroll down and go to matrix and just tick off that.
Yeah, it's this one.
Oh, it's really an old version, 1.6. So maybe you should reinstall it. You can just tick off that, like the tick mark on the left of the matrix.
Yes.
Yes, now you can install it again.
Okay, I have downloaded 1.7. Should I install that one?
Currently, if you just run the line 4, let us see what it installs. Just do yes, it will not really restart.
It says error in installing.
Oh. Okay, it seems that someone has matrix 1.6 and it works, but
I have downloaded the
but the same old versions.
I have this one.
Or you can just type. You can just type matrix there in package circle. Oh, that's fine too. It seems there is some problem because it can't remove the older one.
Oh, I see. So okay, can you tell me what's the error? I'm sorry, they would say.
Oh, sir, it was that it cannot run the function crossprod.
Crossprod x1y. Yes. Okay, so that means it's the parse. So maybe the matrix. I see. Do you have the matrix package installed? Oh yeah, you have it, right?
Yeah, I have it, but I think it's the oldest version.
Okay, is it possible for you to, oh, it's 1.65, it's not that bad.
Yes, like someone with 1.65 had run it successfully, so I can't really figure out.
And you have the latest version of the Bravo, right?
Yeah.
Okay, can we just try that same thing again then? Oh, so you're trying to install the which package? Oh, you're trying to install the matrix package now?
Yeah.
Oh, okay, I see. Is it a Windows machine?
Yeah.
Okay, so that means what you have to do is, so most likely you have to do it manually because, so that's why I think you are not able to uninstall the package. So, what you have to do, you have to go to the, the R package, like basically C program files R, and here you have to manually uninstall it.
Okay.
Yes, so manually delete it basically. Or I don't know whether if you just use the base art, like the art terminal, maybe that will help instead of RStudio. But one way to make sure that you can just go there and manually.
Yes, I will delete it. I will uninstall it.
Yeah, and also I want to just try, like, instead of a list, if you just provide in the SVEN, if you just provide the X matrix, whether it is running.
Yeah, it's running.
Okay, just X matrix is running.
Yes.
Only when he's putting the list, it's not running.
Oh yeah, let us try once again just with the X matrix.
Yeah, and don't use ntemp equal to 10. Just the default ntemp equal to 3 is fine. Then
Oh no, actually, sir, the intent by default is 10 now, I think.
okay, it's 10 now. Okay.
Yep.
Mm-hmm.
You can just put one matrix instead of the list.
And then put ntemp equal to 2 then, or something,
Yeah.
2 or 1.
Yeah, just delete the Z from there. Yeah, and change the lambda to just a 0.1.
Only 0.1.
Yeah, there shouldn't be 2 lambdas.
Yeah.
You don't need to put the C actually. Okay, that's fine. And you can put ntemp equal to 3.
Oh, it can't find the function.
Oh, because maybe the library Bravo was not run.
Okay.
Oh, it's just it's the same problem.
Yes, it's the same problem.
Okay, can 1 minute? Can you just like delete the environment and history, everything?
Which one?
Delete the environment and history, whatever is there, and then run without calling the matrix package. Because there's a crossplot function in the base R package. Maybe that works. So you can just delete like the broom sign. There's a broom sign, the sign of the broom.
Yeah.
That one. Yeah, that one. You can just delete the environment and delete the history too.
Yeah, it's okay. Yeah.
Yeah, so you can delete the history if you want. That will completely refresh your system.
Okay.
Yeah, just do that. And now you can comment out the library metrics and install.packages metrics like lines 4 and 5.
4 and 5, should I?
Yeah, you can just comment them out.
Okay.
But maybe for some other functions, we need that package. Let us see. Maybe not for the crossplot, but for other functions, we might. Someone said that the package metrics and R4.30 is not working together.
I don't think so. But I don't think you can use the base crossplot here because it's a sparse matrix. It's a DCC matrix.
That's true.
It's a DCC matrix. So you can. I don't think that.
Oh, okay.
Yeah.
Because the code we written, we had used the crossplot of the matrix. So.
But so I don't know why is this. Did you try Googling it, Devorsi, the error?
Yeah, I tried.
Okay. Okay, I will try to fix it, and maybe I'm taking your time.
Maybe a safe option will be just to update your R version. Like see if that works.
Okay, or I will restart it again, and maybe that will be. Or I will delete the matrix like manually from the.
Yeah, that will be good.
Okay. I will come back to you if something is.
How do we go VCF file to?
Yeah, how do we go from a VCF file to a sparse matrix? So I often use Tassel to convert a VCF file into numerical genotypes, and then use the numerical genotypes to sparse matrix. So let me do a quick, quick demonstration. So here, let's say I have this matrix, just a toy example. I have this matrix S, which is a numerical genotype matrix, a toy numerical genotype matrix, 4 by 5. So it should be 4 genotypes with 5 SNPs. So toy case. Okay. Now there is a function called matrix with uppercase M inside package matrix with uppercase M. And this is a way to basically convert a matrix into a sparse matrix. So if I say let's say S sparse, if I say call matrix and then on this original matrix S, it will convert into a sparse matrix. How does it look? Well, it looks like this. So only the ones, the basically non-zero values are stored, but zero values are not stored. Now, there's another thing I do. So what happens if you do numerical genotype? I think Tassel, if I remember correctly, Tassel converts the major allele into 2 and minor allele as 0. But that would mean if you code the minor allele as 0, then there are less zeros in your SNP matrix. Instead, we should do convert the major alleles into 0. Now, how do we do that? So let's say if your S matrix, just to look at a toy example, our binomial, let's see, 22. Let's see. Okay, so let's look at this. That's not a good matrix. Yeah, there are some zeros. I mean, okay, let's look at it. So anyway, seems like there are more 2s, right? So I can look at simply 2 minus S. That will give me a lot of zeros. So if you encode your alleles in such a way that the major allele is coded as 2, then you can take simply 2 minus S that will convert the major allele into 0. And then you can call matrix on top of that. Now that would have a smaller number of non-zero values. On the other hand, if you encode your major allele as 1, then you can simply let's look at this particular case. So in this case, the major allele is coded as 1, minor allele is coded as 0. So you see more 1s than 0. So if you simply take 1 minus S, then it flips it, right? So it just codes the minor allele as 1 and major allele as 0.
So, Somak,
yes.
If we look for the first 5 rows and first 5 columns for that variable X you have, which is your sparse matrix, so what does that 0.092? I was thinking I would just get a period, either 1 or 2s or those kind of, yeah.
So these were missing values and they have been imputed. So for genotype 4226, in this particular chromosome, it was a missing. And this is the probability that this particular marker has 9.2% chance that it's a minor allele.
Okay, and the imputation was based on
nearest neighbor and LDDK. So I think if you do it in Tassel, you can get the imputations done.
Oh, okay, thanks.
Yeah, these are we get this from our genetics collaborators. So they are more knowledgeable about.
Yeah, no problem. Yeah, thanks.
So I wonder if there aren't any other questions, if we'd want to end a bit early, or if there is anything else that thought might be useful to share or demonstrate.
Yeah, thanks, Nicole. I think that's a good point. Are there any other questions? Hearing none. So I think it would be a good time to call it a day. And thanks everyone for joining today's workshop. If you have any questions, or if you have any questions, please feel free to reach out to either Dr. Roy or to me, and we'll be very happy to help you. So with that, I hope you have a very nice weekend ahead, and yeah, that's all. Thank you very much. Nicole, I think, yeah, end the meeting, I guess. And thank you, Nicole and Eddie, for, thank you a lot for helping with everything here.
Yes, thank you all for holding this workshop. Very much appreciate it. And just to reiterate, we are recording this. It will be available on the AG2PI website, which is ag2pi.org, on the same page where you probably registered for this event. And again, you will find all recordings of past AG2PI events on the website. So feel free to browse through those as well. And again, thanks everyone for coming, and have a great rest of your day.